Hello everyone, welcome to the ITLS Academy. Today we are going to discuss the topic that is your callus induction. So before starting the lecture, we have to know that what is this callus induction means that. Okay, so before knowing that what is this callus induction, we have to know the definition of this callus. Okay, so callus is what? Just Now, this callus is simply, if we talk about in a plant tissue culture, in a plant tissue culture, this callus is defined as mass of undifferentiated cell that are grown in a disorganized manner. Simply, this callus are what? Callus is a mass of undifferentiated cells and they can be grown in a disorganized manner. Okay, this callus is produced from a X plant. Okay, this callus is produced from a X plant, produced from the X plants. Okay, now these callus are grown on a agar media. Okay, so simply this callus are what? Callus is a defined mass of the undifferentiated cells. Okay, and this undifferentiated cells are grown in a disorganized manner and it is produced by a X plant and can be grown on a agar media. Now this callus induction are defined as a, it is a process. It is a process of creating a mass of undifferentiated plant cell which is called a callus. Undifferentiated plant cell which is called as a callus. Now in today's lecture we are going to see that how this process is work, how this undifferentiated mass of the plant can be grow, which is called a callus. So in this lecture we are going to discuss about it. Now let's start the today's lecture. So before proceeding the lecture, we have to know that the what is this callus? That I already told you this. It is what? It is a mass of the cells which grow unorganized. Mass of the undifferentiated cell which grow in a disorganized manner. Simply. Okay. Now it is what? It is a unspecialized, unorganized, growing and dividing mass of the cells. It is produced when a X plant are cultured on a appropriate solid medium. Okay, here the solid medium are what? It is generally a agar media. It is generally what? It is a generally agar media. So when we culture X plant into a media along with the auxin and cytokinin, which is a growth regulator, in a correct condition, now 2,4-D are commonly used. Then this callus are form. During callus formation, there is a some degree of de-differentiation both in morphology and metabolism, which result in the loss of the ability of the photosynthesis. Okay, so this is about the callus. Callus are what? Unspecialized, unorganized, growing and the dividing mass, and it can be grown by a explant when it is cultured into a media. And in a media, there is a growth regulators are also used. That is your auxin and cytokinin. Okay, and it is what somehow the degree of the de-differentiation in both the term, that is your morphology and metabolism. Morphology and metabolism, which result in the loss of the ability of the photosynthesis. Okay, now the next is, now this callus is what? It is a blob of tissue. Okay, and this blob of tissue are mostly undifferentiated. They are what? They are undifferentiated cells. These callus are naturally developed on a plant as a result of wound. Okay, this callus is what? They are naturally developed into a plant, which is a result of wound. This callus can be left to develop or can be further divided. Okay, this callus can be left to develop or can be further divided. Now the next is your callus culture may be compact or a freeable. Okay, so this callus culture are either compact or either freeable. Okay, here the name suggested that compact means that they will compact like form a compact like structure, like they are very close to each other. And here the freeable callus means that the cells which are associate them, they are loosely packed like this structure. Okay, so that is the difference between the compact callus and the freeable callus. Now see what was written in the slide. Compact show are densely aggregated cells. This freeable callus shows the loosely associated cell, and the callus becomes soft and break apart easily. Now the next term is your habituation. Habituation is, it reduces the requirement of auxin or cytokinin by the culture during a long-term culture. Okay, so habituation is what they reduces the requirement of the growth regulator. Growth regulator, that is your auxin and cytokinin by the culture during a long-term culture. Now, the next is your properties of the callus culture. It is often come from a shoot in early culturing. Okay, so during a early culturing, this callus are grown from a shoot. It can be maintained indefinitely, no photosynthesis, and grow in dark. So, for the growth of this callus, this photosynthesis is not required. So that means that it can be grow into a dark very easily. It can be used to isolate single cell with stem cell-like property, that is your called a totipotent. Okay, so we all know that this callus can also be isolated from a single cell along with the stem cells, and it shows the properties like totipotent. Okay, now the next, materials and method. Now the first method is what? Sterilization of the seed. So we have a mature seed of rice which are cultivator Fujisaka 5 used were obtained from a Mardi, Sebarang, Perai, Pinang. The seed were dehusked and sterilized with a 70% of alcohol for a 10 minute. Okay, so the variety that we choose is a Fuliska 5, which is used for obtained from Mardi, Seabrang, and Pirai and Penang. Okay, and it first of all, what should we do? We dehusk the seed and then it will be sterilized with a 70% of the alcohol for a 10 minute, which is followed by a 30-minute sterilization in 20% of Clorox. Okay, then after that, first of all, what we have to do, we use a 70% of alcohol for a 30 minute. After 30 minute, what we have to do, we use a 20% of Clorox for a sorry, this is for a 10 minute. Alcohol is for a 10-minute sterilization of alcohol with the alcohol for 10 minutes, and then when we use 20% of Clorox, then it should be for a 30 minutes with a few drop of Tipol. Now along with it, what should we use? We use a few drops of Tipol. After rinsing several times in a sterilized distilled water, then this seed were inoculated on a MS. MS is what? Murashaik and Skoog. 1962 supplement with a 2 milligram per liter 2,4-D. Then the culture should be placed. Then the culture were placed in a culture room with a temperature 25 plus minus 2 degrees Celsius. Okay. After that, the growth and the contamination rate were observed every week. Okay, so growth and the contamination rate were observed in every week. There is a three stages of the callus culture. First one is your induction. In induction, what happened? The cells are in a explant. They are differentiated and begin to divide. Then the second stage is your proliferative stage, in which the cell division take place in a rapid way. Then the third stage is your morphogenesis stage. In this stage, there is the differentiation and the formation of the organized structure take place, and especially this process lead to the plant regeneration from our somatic cells. Okay, suppose that this is your somatic cell. Now this somatic cell give rise to a plant regeneration. Okay, now the next is your Callus induction. The first process, callus production from different explant, that is your Oryza sativa LCV Fujiska 5, and the media which is used is MS supplement with 2,4-D. That is the concentration of this MS is 0 to 10 milligram per liter after the 4 week of the culture. Here the explant type, that is your mature seed, root, leaves. Here the 2,4-D milligram per liter. Then the root explant show to be the best explant for a callus formation ability term of weight, but the callus form are highly unorganized. Okay, so after that, the callus which are formed is what they are unorganized, yellowish in color, and differentiate into a root. Now there are two type of callus are formed. One is your embryogenic and another one is your non-embryogenic. Okay, from the embryo scatellum and the embryogenic callus were relatively dry, yellowish in color, compact and nodule in appearance. Okay, if we compare the embryogenic and non-embryogenic callus, then this embryogenic callus are what? They are a dry, yellowish in color, compact in nature, and nodule in appearance. Then the leaves were found to be not suitable as a explant source for a callus induction. Now the next is your callus. This is the three diagram. A represent the mature seed. This is what? This is your mature seed. Okay. B is for what? B is for a root segment of Oriza sativa LCV Fujiska 5 after 4 week of the culture. And the last one is your fine root hair form. Now mature seed were selected as a bed explant for a callus induction of the rice. Now the next is your induction. Here it is the diagram of the induction. Then division. division will take place on here. It is the 8-week growth of the cell over time. 8-week differentiation. Differentiation is either organogenesis or somatic embryogenesis. Then plant regeneration. To the steady effect of 2,4-D and the kinetic combination, approximately 0.5 gram of embryogenic callus of Fujiska 5 were inoculated into a MS medium supplement. Okay, so to see that the effect of this 2,4-D and kinetic combination, we use approximately 0.5 gram of embryogenic callus of Fujiska 5, and this can be inoculated into a MS medium supplemented with a different concentration, like different concentration of the 2,4-D, that is your 0.5 or 1.0 milligram per liter. Then we along with this 2,4-D, we also use a kinetin of different concentration, that is your 0.5 and 1.0 milligram per liter, and it will maintain under a continuous light. And then observation on a callus structure and the color were made every week, and data were taken after 3 weeks of the culture. Then the callus were subculture into a MSPGR, which are free medium plant regeneration. Now the next is your plant regeneration. So now we take time for this. After 3 week of culture in a MS medium which contain 2,4-D and kinetin, then the biomass of the embryogenous callus is increases in all the MS media and kinetin. Then the embryogenic callus remain yellowish in color. Here it is the data between a fresh weight and the media effect of the 2,4-D and a kinetic supplement into a MS or a proliferation of plant Fujiska 5 embryogenic callus. Now the next is, but after the subculture, the embryogenic callus into a MS basal medium, there were some green embryos were detected from all the medium, and only 1 to 2 plantlet bottle were regenerated from these green embryos. It might be indicated that the 2,4-D or a kinetic concentration were increases, and it will need it for a plant regeneration. Now the next is your somatic embryos and complete plant of rice CV Fujiska 5. Plant regeneration of rice CV Fujiska 5. This diagram represents that. Now organogenesis are divided into a two part. Just a minute. Organogenesis are divided into a two part. One is your shoot formation and another one is your root formation. Okay, this root formation are again divided into a one part, that is your shooting, and this shoot formation are divided into one part, that is your rooting. Now this rooting and shooting combine together and form a whole plant. Okay. Now the next is your plant morphogenesis. The first one is your it is divided into two part. One is your organogenesis and another one is your somatogenesis. In organogenesis, different part of different organ of the plant are formed. Different organ of the plants are formed. Now the different organs like your leaf, shoot, roots on plant organ usually of different kind. Now there is the enhancement of the axillary bud, proliferation, development, adventition of shoot formation and adventition of root formation. Now the second is your somatic embryogenesis. In this process, the initiation of the embryo take place. Initiation of the embryo take place, and after initiation there is a development of the embryo will also take place in somatic embryogenesis. and it will be formed from a somatic cells. Okay, now this is about the plant morphogenesis division. Now this organogenesis, again, there is a three form. One is your enhancement of the axillary bud, proliferation and development. There is a stimulation of the shoot apical meristem in vitro that include proliferation lateral bud. Now the second one is your adventitious shoot formation. There is a dedifferentiation or a differentiation and development of the shoot from a non-meristem, either directly or indirectly. Now the last one is your adventitious root formation. There is the initiation in the root adventitiously at the base of the shoot apex, and a vascular continuum is established which complete a plant regeneration. Okay. Now the next is your somatic embryogenesis. Here it is a de dedifferentiation is typically minimal, but a meristomoid-like tissue can be formed in a later case. Okay, histogenesis of somatic embryogenesis is characterized by the formation of the bipolar structure in contrast to adventitious organogenesis. Now, single cell origin of somatic embryo, which make a chimerism infrequent, adventitious shoot, and it can rise from more than one cell. Okay, morphology and characteristic of the callus. Callus tissue are proliferated as more amorphous mass of the cell with a no regular shape. So there is a difficult in describe its external morphology, but they can be discussion on the basis of other characteristics like your texture, coloration, hormone requirement, etc. Now, on the basis of this callus tissue, which are initiate from a X plant of the same plant species, maybe show some considerable variation. Now, texture of the callus are divided into a two part. Either they are soft, either they are hard. Okay. So, as the name suggested, that the soft callus is what they are friable in nature, means that they can loosely packed like this. Okay, they are loosely packed, and it is made up of heterogeneous mass of cell which have a minimum contact. Now this hard scalars are what they are, consist of giant cell, trachea like cell, and they are closely packed and they are compact in nature. Now this hard callus sometimes they have a nodule. Coloration. Generally, callus tissue is creamish yellow or white in color. Sometimes it may be pigmented, and pigmentation are uniform or patchy. Like this is your patchy, and maybe they are uniform. And maybe they are green in color, and sometimes this callus tissue grown under a dark condition, and they will turn into a green color after the transferring into a light. Okay, so this callus are what? This callus when present in a dark, then it will be a white color. Okay, now sometimes this callus should be kept in a light condition, then this white color will convert it into a green color. Some may be yellow in color due to the synthesis of carotenoid pigment. In some coliform culture, it is in purple color due to the accumulation of the anthocyanin in vacuoles. Okay, if we take a example of the coliform culture, then sometime it will appear a purple in color due to there is accumulation of anthocyanin in their vacuole, or even due to the production of the oxide form of 3,4 dehydroxyphenylalanine. Now the next is your, this is a diagram. Again, this is your diagram of the callus. Here you see that it is a yellowish in color, and here it is a purple in color due to the accumulation of the anthocyanin. Now, principle and procedure of this callus culture. So there are three criteria for a callus culture. One is your aseptic preparation of a plant material. So first of all, what we have to do, we have to prepare a plant material aseptic. Now, the second criteria is what we have to select a suitable nutrient media. This is very important because every media is not fulfill the requirement of every type of the plant. Now the third criteria is incubation of culture under a controlled physical condition. Now the next is your aseptic preparation of plant material. First of all, for a aseptic preparation means that the environment free from any type of microorganism. Okay, so first of all, what we have to do, we take a plant and we wash it with a running water 3 hour overnight. Okay, so we wash this plant with our running water either 3 hour or either overnight. Then after that, we use a 1% of Bavistine treatment. Which treatment? Bavistine treatment. Then we use a 70% of alcohol, 1% of sodium hydrochloride, 1% of mercury chloride, and then after that we wash it with the sterile distilled water. So this is your LAF lab. Then next is your selection of a suitable nutrient media. So the concentration for the induction of the root, we required a oxygen and cyanine at the ratio of 10 is to 1 to 100 ratio of 1. Okay, if we take a oxygen 10, then the cytokinin will be at the range of 1. Okay, and it ratio will reaches to 100 to 1. Now if we have to induce the shoot, then the concentration will become opposite. We required at the ratio of 1 is to 10 or 1 is to 100. Intermediate ratio around 1 is to 1 favor a callus growth. Okay, so if the amount of this oxygen and cytokinin become equal, then it will leads to the callus growth. Now we use a agar solidified or a semi-solid nutrient media. The media are autoclave at 15 psi for 15 to 20 minutes at 121 degrees Celsius in a autoclave. Now the next is your incubation of the culture under a controlled physical condition. So for the incubation of the culture, we required a temperature around 25 plus minus 2 degrees Celsius, photo period like 16-hour light and 8-hour light, dark. Okay, so if we give a 16-hour light to the culture, then we have to give a 8-hour dark to the culture. Then the intensity of the light is around 2000 to 3000 lux and the relatively humidity is around 50 to 60%. Now the next is your step involved in a callus culture. So first of all, we take a explant, then Bavistine will be used, then alcohol will be used, then sodium hypochlorite will be used, then uh, mercury chloride will be used, then we cut the explant into a pieces, then the inculation, then the callus formation, suspension culture and new plant. This is how the cycle work. Callus multiplication. Actively growing callus can be initiated culture media with an even physiological balance of the cytokinin and oxygen. After the biomass of the callus, which increases 2 to 4 times after 2 to 4 weeks of the growth, then this callus divided and placed into a fresh media. Suspension culture. This suspension culture can be initiated from any part of the plant and sometimes it is commonly it will initiated from a friable callus which are already grown in a culture, then it will be transferred into a liquid medium. Now the next is your agitation. Agitation means that the breakdown of the cell aggregate into a smaller clumps of the cell, and then the distribution should be uniform and the clumps which are formed in a medium provide a gas exchange. Okay, so this is a suspension significance of the callus culture. This callus culture as such has no major importance unless and until it is used for a other experimental object. Still, callus culture got us some importance, like the whole plant can be regenerated in a large number from a callus tissue. Now, this phenomena is known as a plant regeneration or a organogenesis or a morphogenesis. Callus tissue is good source of genetic variability. Cell suspension culture in moving liquid medium can be initiated from a callus culture. Callus culture is very useful for obtaining the commercial important secondary metabolite. Several biochemical assays can be performed from a callus culture. Okay, so conclusion is that sterilization, hormone concentration, and proper handling is very important in a callus culture for a successful result. These three things are very important: sterilization, hormone concentration, and the proper handling. So these are the references where I take all these data. So this is all about the Callus induction. I hope you will understand. If you have any query, you can follow the ITLS Academy on different platforms like Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn, and subscribe to our channel ITLS Academy on YouTube and get our app from a Google. For you can ask your query to the help that are already slide, that is 7080833450. You can visit our website www.itlsacademy.com. Thank you for watching. Please like, share, and subscribe our channel.